Once upon a time, in a lab not so far away, there were two budding scientists who used Mendel's basic principles of inheritance, which is the backbone of the field of genetics, to create an experiment to figure out how the mutant trait of sepia eyes is inherited and expressed into its offspring. The experiment used fruit flies to see how the sepia-eyed allele, or version of the gene, was passed from parent to offspring. If the allele is dominant, it would be expressed even if the individual only has one copy of that specific allele. If the allele is recessive, it could only be expressed if the individual receives two of the same recessive allele. During the first mating, a male sepia-eyed fly was crossed with a female wild-type eyed fly to produce an F1 progeny with all red eyes, which is seen here. The F2 progeny was generated by crossing two F1 flies and resulted in a 3-to-1 ratio of red-eyed to sepia-eyed flies. This hypothesis, which predicted that sepia eyes was an autosomal recessive trait, was proven true. The results and genetic concepts of this experiment are important and can be applied to all aspects of the medical field. An example of using genetics is in the field of personalized medicine. This movement in medicine would not have occurred without the sound understanding of genetic concepts that are evident in this experiment. The Fly Lab Report Abstract. Welcome to the world of genetics. Genetics, by definition, means a branch of biology that studies heredity, or how characteristics of living things are transferred from one generation to the next. Gregor Mendel, who is also commonly referred to as the father of genetics, is one of the most famous and influential scientists of all time. By performing his experiments through breeding pea plants and observing the traits that each offspring exhibited, Mendel realized that some traits can be passed from the parent generation to the offspring. Although in his lifetime, Mendel's scientific discoveries were not welcome by the scientific community, he is known today as the father of genetics. His basic principles are the backbones of genetics and can be applied to the life sciences in one way or another. The way in which an individual inherits a trait is typically biological. In this example, we will be looking at the inheritance pattern between Simba, who is located on the left, Nala, who is located on the right, and their offspring of Kiara, who is located in the middle. So in every living organism, they are made up of many, many cells. And in each cell, there is a nucleus. The nucleus of every living cell contains a large physical molecule called DNA. DNA stands for deoxyribose nucleic acid. It serves as the instructions or blueprints for the organism to exist and function. DNA often exists in thread-like structures in the nucleus, as seen here. Once these thread-like structures tightly coil around proteins, they are called chromosomes. In a normal human cell, there are 46 chromosomes that make up 23 homologous pairs. In this picture, you can see that there are 46 chromosomes and They are lined up in the 23 pairs, as seen here. 22 of these homologous pairs are the same in both genders and are called autosomes. The 23rd pair are the sex chromosomes because they determine the gender of the species. If an individual's 23rd pair is XX, then the individual is female. And if an individual's 23rd pair is XY, then they are male. An individual's chromosomes is inherited by the individual's biological parents. An individual will inherit half of their genetic information from each parent. So, 23 chromosomes from their mother and 23 from the father. In this example, the 23 chromosomes that were given from this individual's father is in the color pink, and the 23 chromosomes that are given from the individual's mother are in the color blue. A gene is a segment of DNA that is able to code for a specific product, such as a protein. So on this strand of DNA, this portion may be able to code for a protein. So this is called a gene. These products are the physical expression of the gene. Each chromosome has numerous genes located on it. A gene mutation is a permanent alteration in the DNA sequence that makes up a gene. Mutations have the ability to change the way a gene is expressed, which can result in many different versions of the same allele. An example of this is that there are many different hair colors in the human population. These different mutations sometimes have the ability to be passed from the parent to the offspring. This is how mutations can be carried from one generation to the next. There are many different versions that can exist of the same exact gene. Different versions of the same gene are called alleles. However, some alleles are more prevalently displayed and observed in the offspring than others. This is due to whether the allele is dominant or recessive. If an allele is dominant, it will mask the recessive allele and be evident in the progeny. Just because the recessive allele is not being expressed does not mean it is entirely wiped away from the individual. That individual can still pass on the recessive allele on to their offspring. But in order for the offspring to express the recessive trait, they must have received 2 recessive alleles, one from each parent. But if their offspring inherited 1 dominant allele and 1 recessive allele, their offspring will only express the trait of the dominant allele. Each individual will receive 2 different alleles, one from their mom and one from their dad. If we look back to the example mentioned at the beginning of the segment with Simba and Nala and Kiara, let's say Simba is dominant for the red-eye gene, so he's homozygous dominant. And Nala is homozygous recessive for blue eyes. Both of the genes code for eye color, but each has a different allele or version of the gene for eye color. Each individual will receive 1 allele from each parent. So down here, little Kiara will have a big R from her father, Simba, and a little r from her mother, Nala. The genotype of an individual is the sum of the 2 alleles given to the individual. So for Kiara, Her genotype is big R, little r. There are 3 different kinds of genotypes. I've kind of already hinted to them before, which are homozygous dominant, homozygous recessive, and heterozygotes. A homozygous is an individual that contains 2 identical alleles for the same gene. If the individual has 2 dominant alleles, just like Simba, they are referred to as homozygous dominant. And if the 2 alleles of an individual is recessive, then they are referred to as homozygous recessive, just like Nala. If the individual has 1 dominant allele and 1 recessive allele, then they are considered heterozygous for the given trait. This can be seen right here. The phenotype is the physical expression of the trait that can be observed. This is how the body translates a physical molecule to an expression of a trait. So, in this example, Kiara, having 1 dominant allele, will show the allele for the dominant trait and will have red eyes. There are 2 different types of inheritance patterns for traits, which are autosomal and sex-linked inheritance. Autosomal inheritance is the transmission of alleles on the autosomes, so these. Whereas sex-linked traits are passed through the X or Y chromosomes. Punnett squares are a way in which geneticists can predict the likelihood of an offspring of 2 parents inherit a specific trait. With Punnett squares, the viewer looks at the inheritance of 1 trait at a time. To make a Punnett square, you must first make a grid as seen below. Then the genotype of the parents would be placed on the side of the grid. The order of which parent's phenotype goes on which side of the grid does not matter, because they both will yield the same result. So here I'm going to put the genotype big G, and little g. So one parent is homozygous recessive over here, and one is homozygous dominant over here. After the genotype of each parent is placed on the Punnett square, one will then fill in the empty boxes by putting the letters of the genotype that is in the same row and column. So big G, little g, big G, little g, big G, little g, big G, little g. From the results of the Punnett square, one can predict that the ratios of the potential genotypes of the offspring. In this example, there is going to be 100% of the offspring will have the genotype big G, little g, and they're heterozygous. And there you have it. Another influential scientist in the genetics field is Thomas Hunt Morgan. By performing his experiments with the model organism of fruit flies, his students and him discovered classic genetic principles. It was them who also made fruit flies popular as a model organism. One of their major contributions was identifying that the human genome consists of 46 chromosomes in pairs of 23. 22 of the chromosomes are autosomes, or not sex-linked, and one pair is sex chromosomes, which identifies the gender of the offspring. Morgan's team also provided the proof for major genetic concepts such as the chromosomal theory of heredity, genetic linkage, chromosomal crossing over, and non-disjunction. Thomas Hunt Morgan and his team discoveries provided a foundation for genetics. A model organism is an organism that is used due to the fact that it has a certain genetic factor that makes it ideal to study. For ethical reasons, people will not perform an experiment on humans if an animal can be used to receive the same result. Traits that would also make something a desired model organism are that they have a short life cycle, a large number of offspring, the ability to thrive in a lab environment, because they are inexpensive to store. Some examples of model organisms seen here are mice, frogs, plants, worms, flies, yeast, and zebrafish. If you are studying single-celled organisms, the common model organism is bacteria. If you are studying eukaryotic processes, the common model organism is yeast. By studying model organisms, society can find out more about the human body or behavior. In this experiment, the model organism being utilized is the fruit fly. On tracking the inheritance pattern of specific traits. So here we have an image of the fly's life cycle. The life cycle of the housefly starts off or begins in the embryo or egg state, which looks just like a grain of rice. A female housefly is capable of laying up to 150 eggs per laying, which it can produce up to 5 to 6 layings over a period of a few days. The female housefly prefers a damp, dark surface, such as manure, compost, or other decomposing organic materials to lay her eggs. Within a day, the housefly's eggs hatch into larvae, also known as maggots. Maggots are legless white insects that feed from the egg-laying site for 3 to 5 days. During this time, maggots molt several times, and they will choose a dark place to pupate. A fly's pupa is similar to that in function of a butterfly's cocoon. Their hard brown shells protect the inactive developing flies. Over the course of 3 to 6 days, the pupa will transform and develop legs and wings. Once they hatch from their pupa state, they emerge as fully grown houseflies. Within 2 to 3 days, a female housefly is capable of reproduction. So in this diagram, you can see the difference or progression from the embryo state all the way to the adult fruit fly. This one over here is a female, and this one over here is a male. There are multiple ways in identifying the sex of a fly. The major way in determining the sex of the fly is to identify if there is a presence of a sex comb. Sex combs can be found on the front legs of male flies. If a sex comb is present in a fly, it is automatically male, because they do not exist in female flies. So, as you can see here, this is what the sex comb looks like. Another way to sex a fly is to look at the size and shape of the fly's abdomen. In male flies, their abdomens are shorter and rounder than that of the female flies. The last way is to use the abdomen's coloration. In male flies, their abdomen is usually solid black in color, whereas the females have a bee-like stripation to it. Abdomen size and coloration is not a surefire way of determining the gender of the flies, but may have to be used if one is unable to determine whether or not sex combs are present in the flies. In this experiment, we will cross the wild-type red-eyed flies with the mutant sepia-eyed flies. The purpose of this experiment is to figure out how a specific trait is inherited and expressed in the offspring. It is hypothesized that the result in this experiment will follow the rules discovered by Mendel those very many years ago regarding basic heredity and inheritance of genes from parent to offspring. In this experiment, it is hypothesized that sepia eyes was an autosomal trait and will be recessive to the red-eyed wild-type fly. If this hypothesis is accurate, then the following results should occur. Since both the wild-type red-eye fly and the sepia-eye fly in the parent generation was homozygous for the eye color trait, then all of their offspring should be heterozygous and express only the dominant allele for the eye color trait. So if the wild type was the dominant eye color, then 100% of the F1 progeny should be wild type. For the sepia-eyed trait to be an autosomal trait, both the sepia-eyed and the red-eyed trait should be expressed evenly between both genders of the F2 generation. Since the F2 progeny is the offspring of heterozygous parents, then the F2 generation should express a 3-to-1 ratio of red-eyed to sepia-eyed flies. During this experiment, there were 2 different types of mating. During the first mating, 3 female mutant sepia-eye flies were crossed with 3 male wild-type red-eye flies. To do this, a habitat must be created, as seen here, by taking a clear 50 centimeter cubed vial and adding the fly food called Formula 4-24, which is seen here. It kind of has the consistency of instant mashed potatoes. And adding an equal scoop of water. Then a bit of yeast was sprinkled on top of the formula. This will provide the nourishment for the developing flies over the 2-week incubation period. During this 2-week incubation period, the flies must be stored at a 21 degrees Celsius room. Once the habitat is complete, the flies are added to the vial and capped with a white foamy plug, which allows the flies to breathe but keeps the flies in control. So as seen right here, this is the white foamy plug. After 1 week, the parent flies must be removed from the vial. To do this, you must first anesthetize the flies by adding fly nap and allowing them to fall asleep. To do this, you must first dip the absorbent end of the wand into the fly nap. Next, you must take the vial and gently tap it to knock off any flies that may be clinging to the sides of the tube or even to the plug itself. Then, with one finger, push the plug very slightly onto one side. After that, remove the wand from the vial of fly nap and then quickly stick the anesthetic end into the culture vial just below the plug. The fly nap is added to the vial for approximately 1 minute. Over-anesthetizing them will cause them to die, while under-anesthetizing them will cause them to walk around and wake up during working with them. Once the parent flies are anesthetized, then the individual will humanely dispose of them in the morgue, which consists of 100% alcohol. This prevents the parent flies from interbreeding with the F1 progeny. Once the F1 progeny is formed, which will take 2 weeks, one must score the flies, which entails looking at them under a microscope, counting the flies, and determining the gender and eye color of the flies. To score the flies, you must first anesthetize them with a fly nap, and then remove them from their habitat once asleep. Under the microscope, you can properly score the flies. The process of microscopy is, by definition, the use of a microscope to see tiny organisms, or specimens. For this lab, we used a compound microscope to absorb a specimen of flies. This is how you properly use one of these types of microscopes.
Connect it to a power supply. We then set the on-off switch to number 1, this powers up the lamp. What we then do is we make sure that the objective lenses, which rotate on the nose piece, is set to the red lens. That is a low power lens. What we need to do next is take the stage and put it in its lowest position. The slide is then placed on the stage, held in place by the retaining clips. We then raise the stage as high as we can take it. We then look down the eyepiece, and we always focus by moving the slide and the stage away from the objective lens. So while we're looking down there, we will use the focusing wheel and we turn it, and it's about 1/4 turn, and the slide will be focused at that point. If we wish to look at it at a higher magnification, we can rotate the nose piece so that the yellow lens clicks into position. This will give us a higher magnification. There is a third lens here, it is blue, and that must never be used.
Approximately 76 individuals were in the F1 progeny and were generated from this cross. The results were interpreted and recorded. From there, 6 of the F1 progeny was crossed to produce an F2 generation. The 6 flies included 3 females and 3 males. This was done by first creating a habitat and then picking 6 specimens from the F1 progeny, which is seen here. Any additional F1 flies that were not used in the cross to produce the F2 progeny was properly disposed of in the morgue. After a 2-week incubation of the F2 progeny, none of the flies reached adulthood. This required waiting another week for the F2 results. After a 3-week incubation, there were some adults in the F2 progeny. Once the F2 progeny was formed, one must score the flies, which entails them counting the flies, determining their gender, and their eye color under a microscope. 50 individuals, approximately, in the F2 progeny were generated. The results were then recorded and interpreted. In the first mating, 3 male mutant sepia eye flies was crossed with 3 female red-eyed wild-type flies. Approximately 56 individuals in the F1 progeny was generated. Of the total progeny, 100% of the individuals were red-eyed wild-type flies. There were other flies in the F1 progeny that weren't accounted for because their gender could not be determined, but the eye color was still red. This calculates to 100% of the total progeny being wild-type red-eyed flies, as seen here. In the second mating, 3 female and 3 male F1 flies were mated, all of which showed the wild-type red-eyed trait. Approximately 50 individuals of the F2 progeny was generated, but only 43 of the F2 progeny could be accounted for. Of the total 43 progeny, 34 individuals were wild-type red-eyed flies, while the other 9 were sepia-eyed flies. This calculates to a 26.5% of the total progeny had the sepia-eyed trait, and the other 73.5% of the progeny having the red-eyed wild-type trait. This resulted in a 3-to-1 ratio of red-eyed to sepia-eyed flies. The hypothesis stating that the results will align with Mendel's rules of heredity and inheritance of traits from parent to offspring was supported by this experiment. It was also hypothesized that the sepia-eyed trait was autosomal and would be recessive over the wild-type red-eyed flies. This prediction was soundly supported by the results of this experiment. Evidence of this was observed pretty early in the data. To start off, it was known with 100% certainty because it was guaranteed by the company that was utilized for this fly experiment that both the wild-type red-eyed flies and the sepia-eyed flies were homozygous for the trait and was a result of true breeding. So if the wild type was the dominant trait, only wild-type flies would have been expressed in the F1 progeny. That was exactly what was seen in the F1 progeny. All of the flies were red-eyed wild-type flies. Since this was the case in the results, it would be assumed that the wild-type red-eyed fly trait is dominant over the sepia-eyed trait. In addition, since both the sepia-eyed and the red-eyed trait is expressed evenly between both genders in the F2 generation, it can be safe to say it is an autosomal trait. If sepia eyes were sex-linked, one gender would show the trait more frequently than that of the other gender. Since this was not the case in our F2 progeny, it was safe to assume that our trait was autosomal. So, on both accounts, our hypothesis was proven. What was interesting, though, was the ratio that was actually expressed in the F2 progeny. So, if you do the cross, and you bring the alleles down, your expected ratio would be 3-to-1, wild type to sepia-eyed flies. This classic Mendelian 3-to-1 ratio of wild type to sepia-eyed flies was apparent in this experiment. In the F2 progeny, approximately 75% of the flies were wild type, and 25% of the flies were sepia-eyed flies, which results in a classic 3-to-1 ratio. Although this lab was a success, unforeseen events were encountered throughout this process, which may account for minute differences evident in the results. In actuality, we didn't receive exactly a 25 to 75% ratio of sepia-eyed to wild-eyed flies. This could have been caused because not all of the individuals in the F2 progeny was in the adult state. When developing, the ideal temperature for flies is 21 degrees Celsius. Yet over the duration of the F2 development, the temperature fell below the desired amount in the room that the flies were kept. The colder temperatures could have slowed the development of the flies. After 2 weeks of incubation, none of the flies reached adulthood. This required waiting another week for the results of the F2 progeny. After 3 weeks of incubation, there was some of the F2 progeny. There was also a good portion of the F2 progeny that was still in the pupa stage and could not be accounted for in the ratio. Not having the full F2 progeny could have skewed the data. Another reason why the numbers were slightly skewed could have just been that the ratios that were expected is just a probability of what can be seen in the offspring. That does not mean or guarantee that they will be present. Yet even with these setbacks, they did not interfere with the overall results of this experiment. Although not an exact match, the results of this experiment are close enough to view the data as a 3-to-1 ratio of wild-type eyed to sepia-eyed flies. Overall, this lab was a success because the hypothesis was proven and the predicted ratios were observed. The genetic concepts observed in this experiment can be applied to all aspects of the life sciences, especially the medical field. Understanding the working of genetics can better help with the individualized treatment of the patient. Years ago, it was thought to treat every individual with the same symptoms and the same diseases with the exact same treatment. Yet, the one-treatment-fix-all mentality does not work in a majority of the cases, due to the fact that every individual is genetically different. The current movement of personalized medicine is the future of medicine because it looks at the genetics of the individual and then gears the treatment specifically to them. This ultimately increases the success rate of the treatment and allowing the patient to receive the best quality of care possible. This revolution in medicine would not have occurred without the sound understanding of genetic concepts that are evident in this experiment.
That's all, folks.